our national board. She did an amazing job. This is included with your membership. So only members get this. So it might be just a tad incentive. You're kind of on the fence. This would be the time to go ahead and become a member so you can get a copy of this. And let's see. Also, for those of you getting ready to get back on the water, our website has a list of all the whale watch companies. So if you're interested, just pop over onto our website and it has all the information for you. And hopefully we'll see you out on the water soon. So now, what we're really waiting for. Tonight's speaker has spent over 40 years studying bioluminescence. And during our April and May, I guess, event, San Diego was captivated. I know I was. I went out several nights to watch the absolute beautiful blue light that could be seen. So our speaker will share his synopsis of the red tide event, and he will explain some of the science behind these intermittent and sometimes stinky events. And yes, I got the stinky in my house as well, but it was well worth it. So please help me welcome Dr. Michael Latz, marine biologist and bioluminescence expert from Scripps Institution of Oceanography, presenting the Bioluminescence Red Tide of 2020. Thank you.
Well, thank you, Jane, and hello to everyone joining in. I'm going to share my screen and just give a thumbs up if you can see it.
Yes, you're good.
Great. So I'm very excited to talk to you about this red tide event because it's just extraordinary expression of nature. It was beautiful, it was formidable, and it was nauseating towards the end. So for me, I love the bioluminescence. It's something that I've studied my entire career. And usually we do experiments in the lab. So when we have an event like this with beautiful bioluminescence in nature, it's something that's really special. But there's a lot more to this red tide event than just the pretty light show. So what I want to do is first give an overview of the red tide event. Then I'll talk more about the science, and I'll finish up just talking a little bit more about bioluminescence. So I want to start with the basics, and that's plankton. We know that plankton are the numerous microscopic organisms that are abundant in the ocean, and they form the base of the food web. So the phytoplankton harvest the energy of the sun to grow, and they're feasted on, eaten by other plankton, which in turns are eaten by zooplankton, eaten by fish, larger animals, whales. So the base of it are the phytoplankton, and sometimes they can get really abundant. So for this red tide event, the water turned brown. So red tides are not always red, they can be brown or orange. And in this case, there's so many of these organisms in the water that just through millions of cells per liter, they're discoloring the water. So these are photos from Scripps Beach, just showing how spectacular it was. And I'm sure that hopefully many of you had an opportunity to see it during the day. But of course, the spectacular thing was what happened at night. So it was brown water during the day and just this beautiful bioluminescence at night. So it was a little frustrating at first because when this developed, we had the stay-at-home measures, restrictions, the beaches were closed. But when the beaches opened up, then it was a real nice experience to come down to the beach. And this is towards the end of April, and I'm filming from the Scripps Pier, just lucky to see these surfers going out. So for you surfers out there, just imagine what it's like to paddle out through these glowing waves. And when they break, it's like this neon blue light shooting through the wave. And even the paddling of surfing, just getting out to the wave, is stimulating the bioluminescence. Essentially, any agitation of the water is stimulating the organisms to produce light. And I'll talk a little bit later about the science or the ecology behind that. So let's catch one more wave and then we'll move on. So in this case, we have two surfers on one wave. So that's a real treat. Oh, and I have to share this photo. This is by photographer John Moore. And here he captured this image where there's a romantic couple on the beach. There's the moon setting, the waves glowing, and a meteor streaking through the sky.
Wow.
So he considers this photo lucky, but the fact that he was out there for hours, and this is at 3 a.m., I think he just kind of knew what he was doing. So thank you, John, for sharing this image. Spectacular. So we had the beautiful light show, and then things took a turn downhill from there. Towards the beginning of May, we had foam showing up on the beach. And then, so I went out to see it for myself, went out that same night, and there's a lot of foam washing up on the beach. A lot of people out too, checking it out. By the way, this is Hubbs Hall, where I am right now in my cluttered office right here. So I'm very lucky to be able to look out and see the ocean and the red tide when it was occurring. So anyway, I was curious because the foam usually means that the red tide is starting to break down and decay. What was the bioluminescence? So the bottom photo here is beginning of May. This is during the foam period. If you compare it during the peak period, which is the upper photo, you'll notice that bioluminescence was decreased a lot from the peak, but it was still noticeable, still pretty good. And this hung on for several more weeks. So the foam was pretty spectacular. And as we all know, with the foam came the smell. And this was picked up by the media. It had a field day with the headlines. So here's one: neon blue flash gives way to rotting stench as red tide sweeps California coast. Thank you, Los Angeles Times. Fox News: red tide causes big stink along local beaches. And I appreciate from 10 News saying a little nicer: red tide creates strong odor along San Diego's coast. So this is something that was very noticeable, essentially along the entire coastline. It was pervasive and it reached several miles inland. And probably any of you along the coast noticed the stink. So this is due to the red tide degrading, the organisms dying, releasing organic material into the water. That material is feasted on by bacteria that in turn, and other microbes that in turn produce their own metabolites. And those are feasted on by yet other sulfur-reducing bacteria. So they produce hydrogen sulfide, hence the stink. So it was the sulfury smell is the hydrogen sulfide, but also the sewer smell was that, but also there are other organic volatile compounds that were being created as well. So the big question was, when will the stink go away? Now, normally we don't have stinky red tides. So this was pretty bad and it hung on for a couple of weeks. And during this stinky period, what happened is that the levels of dissolved oxygen decreased dramatically. So this is a record from my colleagues at Scripps. They had an oxygen sensor on the Scripps Pier as part of a program that's related to ocean acidification. And so here we're looking at dissolved oxygen as a function of the date. So the red tide is developing here. We get big swings in oxygen because during the day, the organisms are photosynthetic, producing oxygen as a result of photosynthesis. At night, they're respiring, sucking up carbon dioxide. And what happens here, you notice this gray area, Well, this is the zone of low oxygen that's harmful to marine life. Not only did oxygen levels plummet and enter that zone, but there are a couple of days here where the oxygen levels measured at the Scripps Pier were 0. So it's no surprise that we had fish kills. These are sardines along La Jolla shores. There were lobsters and octopus and other benthic animals washing up. And this is a big problem in the enclosed coastal lagoons. Massive fish kills. This is from Agua Hedionda Lagoon in Carlsbad. So the red tide started out really great and beautiful and turned nasty. So now I want to get a little more into the science behind this. And we can formalize the life of a red tide based on different phases. So the first phase is this initiation time. The organisms that are responsible for our local red tides are normally present in our plankton community. They're not invasive species. Sometimes they get really abundant. And in the case of some of them, in this case, they have a life phase called cyst, which is a dormant phase that resides in the sediment, and then they can emerge from the sediment to become swimming cells. So we believe that cysts emerging from the sediment are important in initiating the red tide. But this is offshore, and then the organisms find that the conditions are favorable in terms of sunlight, temperature, and nutrients. I'll talk a little bit more about this. And so they grow, they increase in number, and then they're carried by tides and currents towards the coast. And that's when we see them. We look out, we see brown water, we see patches of brown water, and then that's the first indication that a red tide is occurring. So then there's the termination phase. All good things must come to an end. The organisms run out of nutrients. Their predators grow up and start eating them. And then there's this whole decomposition process involving microbes that I just mentioned. So you may think that at Scripps Institution of Oceanography, we have a sophisticated monitoring center full of computers that's manned 24/7, giving us an indication when a red tide is approaching. Well, no, it's not like that at all. Pretty much, we know a red tide is here when somebody sees it. Now, certainly we have instruments in the water. We can go back and look at those measurements and learn more about it. But the way we know a red tide is here is that somebody sees it. So in the case of this red tide, my story starts with an email that I got from my colleague, Terry Gasterland. And she wrote, "We have a red tide in Del Mar. Early morning pre-dawn surfers report the water glows when they paddle." So they noticed this red tide, not from seeing the organisms, but actually noticing that there was more bioluminescence occurring. So Terry took a sample of the water and looked under a microscope and saw, yeah, a lot of these kind of brownish cells. Well, then the red tide developed. The water really did turn brown, very brown. So Scripps scientists got into gear. There is a shore monitoring program. They took water samples and looked under the microscope, and pretty much the sample is dominated by this one organism. And notice the color, sort of this brownish, tannish color. And that's what's giving this coloring the water. At the same time, there is a plankton camera mounted on the pier that's run by Jules Jaffe. Essentially, it pulls in water and images the organisms in the stream of water under high magnification. So each organism here, this is a single cell, and this is dominated by the red tide organism. So this image from the Scripps plankton camera is from the middle of April, but actually we can go back to the end of March, and this is a different system called an imaging flow cytobot that's moored off of Torrey Pines. And these dark blobs here are the red tide organisms. So this mooring picked up evidence of the red tide approaching the coast at the end of March. So what is it? Well, it's a common dinoflagellate that is usually present here. Dinoflagellate is a type of phytoplankton. They're common members of the plankton community. And for those of you old timers, you may know it as Goniolix polyedra. Yes, so Rick knows that. So it's the same organism, it's just had its name, just renamed the genus. So on the left is a single individual live cell. It contains carotenoid pigments that act as a sunscreen to protect it from ultraviolet light damage, and that's what gives it its color. It also has two flagella, not seen here, but one trails off here, a longitudinal flagellum, and then there's another flagellum within this groove along the equator. So when it swims kind of forward and spiraling as it goes. On the right is a scanning electron micrograph showing that there is a hard structure, part of its skeleton, in a sense. It gives it rigidity. So these organisms are about 35 microns in diameter. Put that in perspective, you could fit about 200 of them on the head of a pin. And so sometimes they just can get really abundant. And we've known about this organism producing local red tides. In fact, Scripps scientists have been monitoring these red tides in La Jolla region since 1900. And so here we are in 2020, and you'll notice that there were fewer of these events that were recorded before the 60s and then the 70s. And since then, we've had a number of these red tide events. They're not all as spectacular as this. For example, 2019, we had a bloom of Lingulodinium lasted 2 weeks. 2018 was 1 week. 2011 was pretty big. That was a month of nice bioluminescence, and there were a number of videos that ended up on YouTube from that event. And that took a couple of months to develop. And then the 90s, there were some really nice red tides. So how does this, the red tide of 2020, compare to some of these other recent red tides? So you can look at some of these main ones, and we can measure the abundance of the red tide organism from water samples. So the maximum for this red tide was 9 million cells per liter. And that's a lot. That's a lot of organisms. So the next highest was 1997, 6 million cells per liter. And then the other red tide events were about 1 million cells per liter. So this red tide event would, at least in terms of the abundance, was had the highest abundance. And I'll make this prediction that it looks like it will be the strongest red tide event on record for this area. It was just that spectacular. So it extended not only throughout San Diego County, but up into the Los Angeles region. And here's some water samples showing really dense brown water from Manhattan Beach and Marina del Rey. And it also extended further south. And this is a remote sensing image. So this is satellite sensing of chlorophyll A, which is the photosynthetic pigment that you can use as a marker for the abundance of this organism. Now, normally there are other phytoplankton that are more abundant, but in this case, the Lingulodinium was so abundant that it was responsible for really high levels of chlorophyll measured from space. And in this image, so here we are, San Diego, the red is red hot, so that's really high levels. This image shows it extending up into Los Angeles, but in fact, it reached all the way up into the Santa Barbara region. And then going far south, there are reports as far south as Acapulco in Mexico. So if in fact the red tide extended continuously from Santa Barbara down to Acapulco, that's a coastal range of 2,200 miles. So that's enormous. That is a really big range for the event. So one thing that was unusual about this time period is that we had a week of heavy rain. In fact, when the red tide was first noticed in that email I got from Del Mar, well, immediately after that, we had a week of heavy rain in that first full week of beginning of April. And I thought, well, that's the end of the red tide, because it definitely is not going to like conditions, rainy and cool, windy conditions. But to my surprise, it actually persisted through that rainfall and then grew. So the question is, did nutrient runoff from the rainfall act to fuel the growth and persistence of the red tide? So we don't have an answer to that question yet. We know that the red tide originated before the rainfall. So the rainfall didn't cause it, but maybe it helped prolong it and strengthen it. So just a few images. This is a cyst of Lingulodinium. So here's a cell, here's the outer covering of the cell. And when conditions are not favorable, it can actually throw off that outer covering and then enter this life history stage of a cyst, which is a dormant stage. So this is a survival mechanism. When conditions are not right, when they're running out of nutrients or there's low noxious conditions, they will form a cyst. This is a non-swimming stage of the cell, and it will sink down into the sediment. And we believe that the cysts act as a seed population to later emerge from the sediment and then repopulate the plankton community, the population. So I mentioned red tides in 95, 96, 97. So it may be, those were exactly pretty much a year apart in the spring. So it could have been that cysts that were created from the 1995 red tide then acted to seed the 96 red tide and the 97 red tide. So I wouldn't be surprised if 1 year from now or 1 year from April, we had a follow-up red tide event. If these cysts emerged and they had this year internal clock for emerging. So it'll be interesting to see if my prediction is anywhere accurate. So I mentioned that there are predators of the organism that become abundant. This is a natural process in ecology where you have a prey organism become abundant and then its predator grows up and eats it. So in this case, this is a predator which is another dinoflagellate, but this is a dinoflagellate that eats. So it's heterotrophic. It's called Protoparidinium. It's also bioluminescent. In this case, these are really interesting because they snare a prey cell, envelop it in this membrane, and then suck out the juices, suck out the organic material from the prey cell and then cast off the whole thing. So these became abundant. And usually we'll get a bloom of this dinoflagellate called Noctiluca scintillans. This is a species that has a worldwide distribution. It is also a heterotroph, so it has to eat, and they can grow very quickly and become very voracious predators on a number of plankton organisms. So turns out, no, they did not form a bloom here in San Diego region, but down in Ensenada, where they had the Lingulodinium, the red tide, then Noctiluca did grow up and create their own reddish red tide. So you notice the color here is a lot more red. So the Noctiluca, they're transparent but they've been eating the red tide organism and picking up the pigment from it, and then they get colored from that. So here we have the predator of the red tide organism forming its own red tide. So that's a very interesting response. And we did have a bloom of Noctiluca locally with the 1995 red tide of Lingulodinium. So this is a photo from my colleague Peter Franks at Scripps. Just for scale, this is a 16-foot Boston whaler. And all this coloration is from the cells of Noctiluca that have been eating the red tide organism. And then essentially here they're starving, they floated to the surface. And this boundary here is not due to the wind, it's just a frontal boundary that they're accumulating at. So the red tide we've been having is considered a harmful algal bloom. And it's harmful, in general, harmful algal blooms can be harmful in several ways. They could be harmful to human health, in that the organisms can produce a toxin that can accumulate in shellfish and lead to paralytic shellfish poisoning and other types of poisoning. And that's what's shown here in this map of these paralytic shellfish poisoning events due to blooms of toxic dinoflagellates. And it's been noticed that there's a lot more of these events that have been occurring. The other feature that seems to be important is that the water column is stratified by temperature. So normally when there's wind, like upwelling winds, the water column is very well mixed so that nutrients are present in the blue zone here. They get mixed up into surface waters, and that is then taken up by phytoplankton. But in the case of our red tide organism, it actually favors calmer conditions when upwelling winds have relaxed and the water becomes stratified by temperature. So nutrients here in the upper layers become depleted by other phytoplankton, and the red tide organism, they're pretty good swimmers. They can actually swim down through the thermocline into cooler water, which is has more nutrients, take up the nutrients, and then swim back up towards the surface. So one of the conditions that seems to be favorable is that it's calmer conditions, relaxation of upwelling winds, and temperature stratification. So it doesn't mean every time that we have a really nice calm conditions, we're going to get a red tide, but this is one of the conditions that appears to be important. And speaking of temperature, the other thing that may be important, we're still not sure, is that this was a warm year in terms of sea surface temperature. So this is a temperature record measured from the Scripps Pier in a time series that goes back to 1916. So the gray area shows, so this is the yearly cycle from January all the way through to the end of the year. And all the gray area is all of the measurements ever taken fit within that range. The dotted line here in the middle is the overall average throughout the year. So it warms up into the summer and then cools down in the fall. So the blue line is the temperature measured at the pier for this year. It's above average. And then towards the end of April, it spiked. And these, the red dots, hopefully you can see, those are record temperatures, the warmest ever measured at the Scripps Pier in this very long time series. So there was warm temperatures. That means that there was stronger stratification by temperature of the water column. And maybe that was important in promoting conditions favorable to the red tide organism. So the other thing, if you remember Al Gore with the inconvenient truth, he talked about the other CO2 issue, that yes, carbon dioxide in the atmosphere is increasing, but a big part of that ends up dissolving in the ocean and changing the chemistry of the ocean. And so we know that levels of carbon dioxide have been increasing. This is the time series, the Keeling curve started by Charles Keeling at Scripps in the 50s, and now continued by his son, Ralph Keeling, continuous measurements showing levels of carbon dioxide in the atmosphere. So we're dumping carbon dioxide in the atmosphere. Some of this ends up dissolving in the ocean, and this can change the chemistry of the ocean. And so there's a program at Scripps that is looking at ocean acidification. One of the things that they're measuring is the pH of the water. They have a sensor on the pier. And so the pH is typically around 8.1. And then what they noticed is that during the red tide, the pH increased. So the water became more alkaline. This is probably a result of all the photosynthetic activity of the red tide organism. But then the pH dropped dramatically during the phase when the red tide was decomposing, when there's a lot of microbial activity. So what does pH have to do with the carbon dioxide? Well, the decomposition involved a lot of bacterial respiration, microbial respiration. And they produced a lot of carbon dioxide, and that carbon dioxide actually dramatically lowered the pH of the ocean. So this red tide event is an excellent demonstration of what happens, this event causing a dramatic, very severe, sharp decrease in pH of the water. So I mentioned low oxygen levels, and here we have another big effect, the much lower pH, much lower than marine organisms would experience. Now at Scripps, in our flow-through aquaria, we had big issues of water quality, and most of the animals in the experimental tanks died. And it's thought that it was more than just oxygen levels, because the aquarius were oxygenating the water with bubbling. So it's thought maybe these low pH levels, because the pH did get very, very low in the aquarium tanks, maybe the low pH had something to do with the very poor water quality that led to the death of the organisms. So there's more to the science. This is just a first look, and it takes a while to work it all up, just to give you a little bit, a sense of the science. So just to talk a little bit more about bioluminescence, because after all, that's my big love. There's dinoflagellates are, not all dinoflagellates are bioluminescent, and there are other groups of organisms that are important sources of bioluminescence in the ocean. So this is here at the upper left is a petri dish in which there are glowing bacteria that are plated. A lot of bacteria in the ocean are bioluminescent. You know how you see underwater footage with all the little specks of marine snow floating around? Well, if you could really turn off the lights and had a very sensitive camera, what you'd see is that most of those specks are glowing because they're colonized by bioluminescent bacteria. So there's many types of bioluminescent zooplankton. This is showing a deep sea copepod called Gaussia, and it spews out bioluminescence to protect itself from predators. There are a lot of jellyfish, especially in the deep sea. Most of the jellyfish in the deep sea are bioluminescent. They'll use it as a predator defense. And in the case of this medusa called Colobonema, it has these tentacles, and when it's attacked or disturbed by a predator, it will autotomize the tentacles, cast them off. The tentacles will kind of writhe around like a worm and be glowing, and that will distract the predator from the medusa, which they can then swim away. So there's a number of polychaete worms that are bioluminescent. This is a deep sea worm, a tomopterid worm. And this is showing bioluminescence from a psyllid worm, a polychaete that lives in places such as San Diego Bay. So, Mission Bay. So you can go to Mission Bay in the summer, and a little bit after the full moon. These worms are bottom living. They live in parchment tubes on the bottom, but just they have this monthly cycle where the females will swim up to the surface and they spew out their gametes in this glowing cloud of mucus. The males have big eyes. They see that glowing mucus and they come up and then they fertilize the eggs. So the bioluminescence used is a cue for these mating swarms. So here's an anglerfish. We all know about them as they culture bacteria here in this modified fin ray called an esca, and the bacteria act as a glowing lure to attract prey to them. So they're ambush predators. You notice the eye looking up here. When prey gets here, then they just pivot and strike. So they're using bioluminescence to attract prey to them. These are flashlight fish. They also culture bacteria in these organs below the eye, and they use the light produced by the bacteria as a flashlight. So they come out, they live on dark reefs, and they come out at night and use the light to search for food. Then here, lower left, this is dim glowing bioluminescence produced by a squid that acts as camouflage. So this is a ventral view. We're looking underneath the squid, looking up. And this is a problem for animals that live not in the deep, dark ocean, but kind of in the mid depths, where there is some light that downwells through the water. It's pretty dim, but animals there are vulnerable to predators looking up and scanning for their silhouettes. So what they do to protect themselves from those predators is to disrupt their silhouette with bioluminescence. So there's squid, there's some fish that do it, and for my graduate research, I studied a shrimp and showed that it uses its dim glowing bioluminescence to mask itself, to act as bioluminescent countershading of the body. So these are all pelagic or swimming organisms, and they're also benthic animals. This is showing a brittle star where the arms are lighting up, and they produce their bioluminescence, protect themselves from predators. They can autotomize an arm, and so a predator will grab an arm, brittle star can release the arm, and then the predator ends up with an arm, and then the brittle star crawls away and they can regrow the arm. So just give you an idea of some of the other functions of bioluminescence. What they all have in common is that the light is produced from a chemical reaction. And so there is a substrate molecule that's called luciferin, and this is a molecule that will be oxidized. So in the presence of oxygen, it will release energy, and this energy comes out as visible light. This chemical reaction is facilitated by a protein called luciferase, and that is the catalyst. It has a binding site for the luciferin, and so it's that binding that controls the light reaction. So there are different classes of luciferins and their luciferases. So for example, a dinoflagellate luciferin and luciferase are different from a squid luciferin and luciferase, different from bacterial luciferin and luciferase, and so forth. So there are different chemical families. There are a lot more to be discovered and characterized, but they all the light comes out from a chemical reaction, releasing energy as light. And the most common color for the light is blue-green. That's the color of light that transmits best through ocean water. So the organisms are producing light for a reason, and that is to affect the behavior of whatever animal is seeing that light, whether it's to attract potential prey, scare off predators, or attract conspecifics for mating. So the chemical reaction is kind of like analogous to a glow stick, but there are different chemicals in a glow stick. One of them starts off in a vial. That's why you have to crack the glow stick, allow the chemicals to mix, and then you get light released from a chemical reaction. It's an oxidation of the substrate molecule. So, and you can tune the color of the light based on the chemistry. So it's kind of the same way in bioluminescence. There's this chemical reaction inside a living organism. In the case of dinoflagellates, the light, so they're single cells, they're microscopic. The light comes from individual sources within the cell. So this is a large dinoflagellate called Pyrocystis. And maybe you could see there are all these little spots within the cell. So those are vesicles that contain the luminescent chemistry and are producing the bioluminescence. So I want to show you this video. This is taken by artist Yvonne Koo. And Yvonne was the one worked with me on the Infinity Cube exhibit at the Birch Aquarium. You may have had an opportunity to see when it was on display. So here we are in slow motion, but these are individual unrestrained dinoflagellate cells that are experiencing a mechanical force and are lighting up. And here we are at higher magnification. So you can notice all these individual sources within the cell, and maybe you'll see there's a dark spot in the center of the cell. Well, that's the nucleus. So the nucleus, they're not producing light from the nucleus, just around other parts of the cell. So the dinoflagellates are feeling a force, and then they light up almost instantaneously with the bioluminescence. And they do this for predator defense. So imagine a scenario, say the Scripps Pier, and it's nighttime. So there's a whole bunch of dinoflagellate cells in the water. And here this represents a zooplankton predator of the dinoflagellate. It doesn't see the dinoflagellate to try to capture it. It's a grazer. It swims around and hopes to grab it. And then you have this fish here, which represents a visual predator of the zooplankton. So this fish predator would love to capture that copepod, but it can't see it because it's dark. So imagine a scenario, all three of those are together. Copepod, the zooplankton swimming around, it bumps into a dinoflagellate cell, grabs it. That force is enough to make the dinoflagellate cell flash. It produces the flash of light, which lasts about 10 milliseconds. Well, 100 milliseconds, so a tenth of a second. And that is enough for this fish predator to see and then come in and try to eat the zooplankton predator of the dinoflagellate. So what's interesting is, what does the zooplankton predator do? Well, it reacts. It has a response. It does this jump behavior that it will jump and try to swim out of there. Because a copepod or zooplankton that doesn't do that is more vulnerable to being eaten by its fish predator. So in a sense, this whole scenario is like a burglar alarm. In this case, the alarm is a flash of light given by the prey, the dinoflagellate. The zooplankton predator is the burglar, and it's setting off this alarm, and it's making itself vulnerable to the fish predator, which in this case is like the police. So the police, in this case, want to eat the burglar. So for the burglars that don't care about the alarm, just like a burglar maybe trying to break into our house, those burglars are more apt to be caught by the police. In the same way here, the zooplankton, the predators of the dinoflagellate that don't react to the flash are more apt to be eaten. So when you have a large number of dinoflagellates present in the water, any motion can set off their bioluminescence. So you can think of it almost like a minefield, that any motion is going to stimulate the light. So here's a video showing lobsters in a tank with dinoflagellates, and just the flicking of the antennae are stimulating the cells. And here's some light. Oh, it's stimulated by a shrimp in a tank. It's giving away the position of the shrimp. And here comes a squid predator homing in on its prey based on the bioluminescence that the shrimp was stimulating by the dinoflagellates around it. So this is a real problem, that if you have an animal moving around in this soup of dinoflagellates, it gives away its position to the predators. So essentially, any kind of motion or agitation of the water is going to stimulate the bioluminescence. During a red tide, it's just really spectacular. And this video is exceptional. And this is by a whale watching group out of Newport Beach. They went on their boat and they just happened to come across dolphins that were swimming the bow of the boat. This is probably like some of the best bioluminescence footage I've ever seen. Now, the dolphins, of course, are not bioluminescent, but they're swimming. The forces associated with their swimming are lighting up the dinoflagellates during the red tide. So this is really incredible. And so this is actually, well, first of all, this is something that has been captured in art, that the artist M.C. Escher was traveling. The story is he was traveling in Southern California in the early 1920s during a time of bright bioluminescence. Maybe it was a red tide. And he went out on a boat, and this is his artistic representation of dolphins swimming at the bow of a boat. So this is a woodcut print entitled Dolphins. And in my own research, we have used the dinoflagellates, the red tide organisms, to visualize the flow around a moving dolphin. So we're interested in the boundary layer around the dolphin. We know that dolphins are streamlined, but we wanted to study this more experimentally, but you can't take a dolphin and swim it in a water tunnel. So we tried working with wild dolphins, but unlike the Newport Beach video, we couldn't get them to come to our boat. So we ended up working with trained Navy dolphins, swimming along the boat offshore at night during a red tide. And so we got images. This is with a high resolution scientific grade camera, so it's in black and white. And we could model the shape of the dolphin as an ellipsoid. So here you have a very thin boundary layer, very thin friction layer at this initial part of the body, just like here. Very little bioluminescence stimulated in the upper part of the dolphin. But then when you have structures that disrupt the flow and cause flow separation, like coming off the fins, off the blowhole, you get more bioluminescence. And that's what we saw here with an ellipsoid. If we trip the flow, if we had here is an O-ring to disrupt the flow, then we get here a thicker boundary layer, and this is nicely visualized based on the bioluminescence. So we could study the hydrodynamics of a moving dolphin quantitatively and model it based on an ellipsoid. And this has been published a while ago, if you're interested in looking at that. So I came to Scripps in the early 1990s. Didn't actually look like this. This photo is in its very early years in 1904. Here's the old Scripps building, still standing, historic building. So I came, it was a little more built out, but one of the really amazing things is my first red tide in 1995 made a big impression on me. This was the same organism. It was very abundant. We had the brown water during the day, and then the spectacular bioluminescence at night. So this was a strong, very strong red tide event, but we didn't have the big stink. We didn't have the fish kills, not as strong and pervasive as the one we've just had. But what was interesting is that the America's Cup was in San Diego during that time. I don't know if you remember it, but they were filming the red tide. They're filming the red tide. They're filming aerial filming of the America's Cup. And here are two boats sailing through the red patches of the red tide that you can see as these brownish patches. And you see the wakes of the boats nicely shown here because they're sailing through the red tide, which is very close to the surface of the water. So it's nicely demonstrated by them cutting through these patches of the red tide. So the red tide, it's not always the water is evenly brown, but when you look a little offshore, it looks patchy like this. Certainly, as it's brought onshore by tides and currents, it will accumulate more along the shoreline. So just in my own research, I'm very interested in this process of the agitation of the water, how that stimulates bioluminescence. So we're looking at how forces act on individual cells, and we quantify these forces, and then looking at the biochemistry, the biochemical steps within the cell that lead to a flash of light. So the time from stimulation of a cell, mechanical stimulation of the cell, to the light emission is only 15 milliseconds. So it's a very, very rapid response. And I also use bioluminescence as an educational tool. That I work closely with the Birch Aquarium at Scripps, and we have like these little packets of bioluminescence, excellent educational tools. I've worked with conservation groups in Puerto Rico to study bioluminescent bays. Imagine a place where you can go where red tides essentially are occurring year round. So these are bioluminescent bays in the Caribbean. And even with one of the conservation groups, we created a salon day of bioluminescence with live materials. And so they bring tour groups through to teach them about bioluminescence. And then last thing I wanted to mention was Infinity Cube. So this is a bioluminescence art exhibit at the Birch Aquarium. Hopefully several of you have had an opportunity to see it. So this was a collaboration with artist Yvonne Koo, and she had come. I have an artist in residence program where I will invite artists to come into my lab and use my resources and imaging equipment to do projects with photography and videography involving bioluminescence. So I've had artists work with like bubbles and bioluminescence and other forms of agitation, even sound stimulation of bioluminescence. In the case of Yvonne, we did the flow. I showed some of the close-up videography she did. And so she did really incredible videos of bioluminescence that were then projected onto the walls of this reflective cube, Infinity Cube. It was this exhibit at the Birch Aquarium 2017 and 2018. So these art collaborations are a nice way to extend essentially the continuum of how I can communicate my research. That certainly when I talk about bioluminescence, I can use lots of jargon and technical terms, but there's this aesthetic beauty that we're all aware of, and the artists are really good for showing that. So they bring a fresh approach. And so we had this bioluminescence art exhibit at the aquarium with none of these technical terms. It was just the imagery and other graphics. In fact, we use haiku as 3-line Japanese form of poetry as a way to communicate the science concepts. So it's a very innovative exhibit. So that's kind of an overview of this red tide and bioluminescence. Here I am. I feel really lucky to be at Scripps. It's a really amazing place. And There's people, my colleagues, doing all kinds of research. And so in the case of the red tide, they're not necessarily studying a red tide, but they have instruments in the water that provide information that help us put together a story about the conditions that were present during the red tide and allow us to understand better the dynamics of this really incredible event. So thanks very much for sharing.
Great job, Michael.
And answer any.
Will you answer a few questions? I've been collecting them in the chat room and I'll ask them. Is that okay? A few questions?
Absolutely.
Gina asked early, and these will go in chronological order. So Gina asked, is it safe to swim in a red tide?
So the answer is yes and no. So there are some people who are sensitive to aerosols produced from this red tide. So most people, during the healthy part of the red tide, it's not harmful. And there are some red tide dinoflagellates that produce toxins that can accumulate in shellfish. The dinoflagellate that forms Florida red tides are toxic, and they produce a compound that becomes aerosolized, blows onto the beach, can cause respiratory issues. So this Lingulodinium is not that kind of harmful species. That there are some strains that can produce a toxin, but the local strains are not known to be toxic. Certainly swimming in it and even getting in your mouth is not going to be harmful, but there are some individuals who are sensitive to the aerosols and will have respiratory issues. So obviously those people should stay away. But when the red tide got nasty and started decaying, there were a lot of there are materials in the water, and then I would not recommend going in during that phase, during the foam phase, because there were issues of like swimmers reporting rashes.
Yeah.
It was a problem. So,
before the deterioration, how would one know, other than an exposure and getting a rash, that they were among the sensitive group?
Well, if you're sensitive based on the respiratory issues, you're going to know when you go to the beach.
Okay.
Like I have colleagues, and they can just walk into a room where there are cultures growing. We can culture these in the laboratory, and they're just the aerosols, the volatiles produced, they know. So if they have asthma-like condition,
right,
wheezing, then they're sensitive and they should stay away.
Okay.
But so my rule, if you're asking what I would do, then if I see foam, that means there's a lot of organic material. The foam is due to the protein. So it means that cells are broken open. There's a lot of protein, and the protein is foaming. It's exactly the same process as beating egg whites. That, egg whites are the protein albumin, and you beat it, you're essentially getting air bubbles mixed into it. And as the protein breaks apart, the parts of it coat the air bubbles and stabilize the air bubbles.
Yeah.
That's the exact same thing going on in the red tide during the foam phase.
Excellent.
So if it's foamy, I'd stay out of the water.
Okay. Laura wants to know, other than global warming, could red tides also be caused by fertilizer runoff? And you were sort of hinting at that with the rain.
Yeah, so there are some red tides and other harmful algal blooms that are definitely fueled by nutrients. And in terms of eutrophic conditions, a lot of nitrogen and phosphorus entering the water. These red tides, in general, if there's a lot of nutrients entering the water, there are other phytoplankton that grow much faster than these dinoflagellates that will use those nutrients. So in the case of these dinoflagellates, certainly maybe the nutrients are prolonging the red tide, as I mentioned, but I think they're using, it's not a simple runoff of nutrients, that it's a little more complex than that. And it may be that they need the water, the chemical state of the water needs to be conditioned in a way from other phytoplankton occurring first to condition the water in the right way. So the chemistry, we're still unclear about exactly what's going on.
Very good. Our board member, George, says that he checked Mission Bay a couple of times during the recent red tide, but did not see any glowing on nights when he did see it on the ocean side. Any idea why that might be?
It could be that the red tide was still hugging the coast and maybe just hadn't entered there. I guess I'm not familiar with what the exchange of Mission Bay is, how strong the tides were at that point, so what the exchange was. But certainly for other red tides, Mission Bay has gotten very bright. So it just may have been the phase of the moon, or I don't know, or maybe at that location offshore that it was staying offshore and not coming on close enough to enter the bay.
And George also wants to know, and this kind of relates back to the Cetacean Society, are baleen whales known to use bioluminescent clues to guide their predation, similar to what you described for fish and cephalopods?
That's really interesting. I'd have to say the answer is, I hate to say I don't know, but I can't make up an answer, but
that's why we're in science. It's because we don't know we're in science. So
exactly. So that's a great hypothesis that the baleen whales would use the bioluminescence to find their prey. I love that.
And then Gina wants to know, how large are the cysts? When you agitate the dinoflagellates to incite bioluminescence, can you do it too much and exhaust these creatures' ability to do so?
Yeah, so the cysts are, this one cell forms one cyst. So it's a little bit smaller because they're throwing off these outer cell membrane layers. So they'll be maybe 25, 30 microns across. They're non-swimming, they sink down. And certainly, yeah, if you agitate them, if you disturb them too much, like even in the laboratory, if I shake them up too much, then they are not going to like that. They're going to form cysts. But actually, an easier way than standing around shaking them up is that you can centrifuge them, they'll form cysts. You can actually put them in the freezer and chill them down. You put them in the refrigerator for a day, they will form cysts. So they are trying to survive. Yeah. So you can do different things to them and they will form cysts.
And we have 3 more questions if you have the strength. If you're still glowing, you seem to be still glowing.
I'm good. Yeah, I love questions.
You haven't reverted back to a cyst yet. So Lee wants to know, are there any plans to use bioluminescence in non-science areas for development in benefiting society?
Yes, and it's already occurring, but I'm not doing it, obviously. So let's say if you did a search of the scientific literature for bioluminescence, and you would find that there's orders of magnitude more publications on the applications of bioluminescence than there are on the ecological aspect of bioluminescence. So for example, the chemistry. So I mentioned luciferin, that is the substrate molecule, and luciferase, the protein. Well, there are a number of luciferases and their sequences are now known. And so you can use bioluminescence as a way to, for example, to look at gene expression. So in the era of bioluminescence, you can clone in the sequence for the luciferase gene. You clone that into a promoter site of whatever gene you're interested in looking at. When the gene is turned on, it'll make luciferase as well. And then you can, in the presence of luciferin, that matching luciferin, you'll get light. And the same way it's been done with molecule green fluorescent protein. So this is a protein from the bioluminescent system of a jellyfish. And it's probably one of the most important tools in cell biology, that it's been used to look at gene expression, and it's been used to look at protein activity. And again, in the same way that its sequence is known, you can clone it and use it as a very powerful tool. Green fluorescent protein it's so important that there are 3 scientists who worked on it. One discovered it, the second cloned it, used it as a tool, and the third made it even better by making synthetic green fluorescent protein, not only green fluorescent, but other colors of fluorescence. Those 3 scientists were awarded the Nobel Prize in Chemistry in 2008.
Wow.
And one of them was Roger Sen, who was a chemistry professor here at UC San Diego. So
Very cool. Very cool. It's obvious that you walked from your building over to that old PRL building that you and I were discussing before the meeting started, talking to those people that are doing all that. 2 more. Are the toxic algae blooms in fresh water similar?
So those are, a lot of those are caused by they're phytoplankton, but they're cyanobacteria. So they're actually photosynthetic bacteria, and they produce, so they're not dinoflagellates. Dinoflagellates are eukaryotic, so they're not bacterial. And so those are photosynthetic bacteria, and they produce a toxin called microcystin. And that is very dangerous for drinking water supplies. So I'd say it's different in that those blooms are fueled by nutrient runoff. So those are eutrophic events, and it's a big problem for drinking water supplies. But I would consider those separate from the marine, the ocean harmful red tides, that they're not just dinoflagellates, but also there's a harmful species of diatom off the coast of the US West Coast that causes problems for marine mammals and other ocean life. So I consider them different, but along the spectrum of harmful algal events.
Very good. And then Barbara wants to know, why are sometimes these organisms coming more up, or are a lot more than usual to be seen? Well, what is the cause? Do we know?
Yeah. So I showed the map of 30 years from 1970. There were a lot less events for the paralytic shellfish poisoning than now. So the question is, are we just getting better at monitoring them and sampling them, or are there actually more events? So it appears to be kind of a combination of the two. We are a lot better at surveying the ocean, especially coastal areas. For example, the state of Florida has a massive monitoring program because along the west coast of Florida, the red tides are such a problem with these respiratory issues, fish kills, even manatee deaths. And so there's big problem in shellfish poisoning. So it's big problems in Puget Sound and New England. So there's a lot more monitoring going on now. In terms of they're fueled by nutrients, the nutrient runoff certainly can fuel them, but a number of these events do start offshore. So there just may be related more to the climate, the global climate changing rather than nutrients per se. But I would say that with in this case, with a warming climate, a warming ocean that increases stratification, in general, that's going to reduce the amount of mixing of nutrients into the water, upper ocean, that'll reduce the production by phytoplankton. So there should be less phytoplankton in general, but it will promote more the dinoflagellates that can deal with these strong thermoclines. So there are changes occurring in the ocean as a result of, in this case, the warming climate.
Well, very good, Michael. And I'm going to throw it back to Jane in just a second, but I want to thank you very much. I want to acknowledge that Dr. Quayman is bioluminescing on my screen right below you. Yeah. He is a faded light. So there's my mentor, and we talked poorly about him earlier, so don't tell him what I told you about. And it's back to you, Jane. Hi, Jerry. Back to you, Jane.
Hi, thank you so much. Everyone, it's been a pleasure. Thank you so much for joining us, and we will look forward to seeing you next month. And please be safe out there. Thank you so much.
And we are recording these and are going to make them available. So
Okay.
Thank you.
Thank you.
Thanks again, Michael. Thank you.
Thank you, Michael, very much.
everybody. You're welcome. Bye.
I'm trying to leave. Let's see.